Today we're going to talk about plants and how they transport water through them. Now, if we imagine this is a plant that I've drawn here, the leaves are waterproof. We've learnt that they have a waxy cuticle on them, so they're not going to take in water through the leaves. In fact, the water is taken in through the roots because there'll be some water in the soil here. And so water will enter through the root hair cells by osmosis. And it will bring things like nitrates, phosphate, calcium, magnesium, and so on as well. So we call those things together minerals. So it's absorbed by osmosis into the root hair cells, into the roots, and then it travels through hollow tubes called xylem vessels up through the stem and out to the leaves. And obviously I'm not going to draw every single vessel there. Now, that's important because the leaves need water for photosynthesis. Okay, so going to jot a few things here. The leaves need water for photosynthesis. You remember the word equation, glucose plus, the word equation is carbon dioxide plus water, and then an arrow with light on it. That gives you glucose and oxygen. So we need water to be brought in through the roots and up through the stem and out to the leaves for photosynthesis. As we mentioned, the movement of water also moves minerals, and so we need calcium to make new cell walls, we need nitrates to make proteins, we need phosphates and so on, and all sorts of minerals to make the structures that we normally find inside cells like chloroplasts and so on. As water passes up through the stem, well, we learnt about cells being turgid and how if they're full of water, they're turgid, they can give support to the stem. And we talked about if we forget to water the plants, they start to wilt because they've lost that turgor. So water passing through the plants gives support. And finally, a lot of the water that actually passes through the plant is lost from the leaves as water vapour. And that process is called transpiration. And that's what we're going to focus on today. Now, if we look at a diagram section through a leaf, so if we were to take a leaf and cut through it, we would cut through the leaf, you're going to see a very thin layer, and what we've done here is just take a little cross section of that. So, a little bit of revision. These cells here make up the upper epidermis, and these ones here are the lower epidermis. And then we've got palisade mesophyll cells making up this tissue here, and spongy mesophyll is the name of this layer here because it's got lots of big air spaces. Now, a few other labels we could put on. That's the waxy cuticle. And there's also a waxy cuticle on the lower surface as well. So, water gets in to the plant through the root hair cells, comes up through these vessels, and eventually it makes its way out through these vessels into the leaf. So we have lots of water molecules inside the vessel. The water can move into the neighbouring cells by osmosis. So some will go up into the palisade cells for photosynthesis, but a lot will go down into spongy mesophyll cells. That make means you'll get a layer of moisture lining those spongy mesophyll cells. And, well, if you've ever spilt a little bit of water onto a surface and you've tried to wipe it up, it's very hard to get it all wiped up. So quite often you leave a little bit of moisture behind. But if you come back in an hour's time, you'll notice that it's disappeared, and that's because it's evaporated. You don't need really hot temperatures to evaporate water. If there's a little bit of heat, yeah, it'll disappear. So what happens here, these surfaces get moist, and then whenever the sun rises, the temperature increases a little bit, the water will start to evaporate, so you'll get lots of water vapor molecules inside, and that's inside the air space. Now, the air space is immediately above this gap here. So this is a gap. It's called a stoma. Plural is stomata, and you're probably more familiar with that word. So it's a gap. Now, if I just rub out the red pen there, you can see that there's a high concentration of water vapor molecules inside the airspace, but there's nothing outside in the air outside. So high concentration to low concentration, diffusion takes place. Water molecules move from a region of high concentration to a region of low concentration until both concentrations are equal. So that's diffusion. And that whole process is called transpiration. So there's really two steps in it there. First, where the water is evaporated from the mesophyll cells. And the second step is where the water vapour diffuses out of the leaf through the stomata. So our definition there, you can see that we've got a stoma on the lower surface of the leaf. The vast majority of them are on the lower surface of the leaf. There's only a handful of them on the upper surface. Back to our diagram here, we could label these two cells here as guard cells. Now, I do hope your diagram is a lot neater than mine. So at night time, these guard cells actually change shape and they will close over the pore. So the stomata are closed at night. But during daylight hours, that's whenever then they start to, during daylight hours, then they'll open up the stomata. And so the pores are open again. So they're closed at night, open during daylight. In our books here is at the top of page 14 in my book. Now, it might be 14 in your book, just depends how you labelled it. But transpiration is the evaporation of water from the mesophyll cells. That's the first bit. And then when it evaporates into the airspace, then it diffuses out through the air spaces and the stomata. So that's how water is lost from the plant through those little pores called stomata. Now, stomata on the lower surface. Stomata, as we said, they're small pores. They're found on the lower surface mainly of the leaf. And the two cells that are either side of the stomata are called guard cells. Now, what kind of factors would affect transpiration? Well, transpiration, you're trying to evaporate water, and then you want to the water vapour molecules to diffuse away. So if you think about trying to put washing out in the line, you want a warm day, and you probably want a windy day. And another thing, you definitely want a dry day. So if you're going to put your washing out, those are the ideal conditions. So those are the ideal environmental factors. And it's pretty similar in transpiration. Transpiration will happen fastest whenever temperature is high, whenever the wind speed is high, and whenever it's a dry day. Now, we don't just mean that it's not raining. Humidity is a term that you might hear sometimes on the weather forecast in the summer's day. High humidity describes a day where there's a lot of water vapour in the air. Low humidity is a very dry day. In other words, the air has no water vapour or very little water vapour in it. So sometimes in the summer you get a very humid day. Some countries are very humid in the summer. So Florida, if you've been there, you'll notice that there's a lot of condensation. You might sweat a lot. And that's because there's so much water vapour already in the air that it makes evaporation of your sweat very difficult. So plants are affected by humidity as well. The higher the humidity, the slower transpiration will be. And that's because a very humid day would already have a lot of those spongy mesophyll cells. So if we just think of the lower epidermis, and we'll put a guard cell here and here. And we've got all our spongy mesophyll cells up and around here. So if this bit becomes moist in the airspace and we get lots of water vapour molecules evaporating into here, if it's a dry day, there's a steep concentration gradient. We're comparing here, hold on, with here. So in that case, there's a steep concentration gradient. If you compare that to of water molecules in the atmosphere, and now you can see that the concentration gradient isn't just as steep. The two concentrations are more similar, so there's going to be less diffusion occurring, or diffusion will happen a lot slower. So on a very humid day, you can see that the concentrations are almost the same. So transpiration is going to be even slower. Now, we can actually look at or try and work out the rate of transpiration by using apparatus like this. Now, there's a bubble in this apparatus. So this apparatus is called the bubble potometer. The word meter means it's measuring something. So what are we going to measure? We're going to measure the distance that the air bubble moves along the scale. So when we're setting this apparatus up, it's really difficult to set it up because there's lots of joints here, and we need to be very careful and make sure that everything is watertight and airtight, that there's no leakages, because leaks are the main difficulty with this apparatus. So we set it up underwater. Now, that doesn't mean that you get a wetsuit on and you dive down into a swimming pool. It simply means that you fill up a basin with water and you submerge the tubes that make up the potometer. So they're underneath the water. Then you take part of a plant and you cut a leafy shoot off it underwater and insert it into the rubber tubing here. And then you would normally put a bit of wire around there, tighten it up, and take the apparatus out. So everything is full of water. It's all been set up underwater. So that's really important. Now, I don't know why that keeps jumping around there. So under water. And another thing, you would normally get some kind of Vaseline or petroleum jelly and just put it around all the joints there. You would just rub it over there to make sure that it's all airtight. Now, how does this work? Well, we've got our leafy shoot, and as time goes on, water's going to evaporate from the lower surface of the leaf by transpiration, and that's going to cause water to be sucked up or absorbed in through the bottom of the leafy shoot there. So as water moves along there, that's going to move that bubble towards the plant. Now, rather than say to the left or to the right, it's easier if you think of it moving towards the plant because it's a plant that's really sort of sucking it up. So what we need to do is record the initial position of the bubble and the final position of the bubble. So that we can calculate the distance moved by the bubble in a particular period of time. Now, that gives us then a measurement of the rate of water uptake. So that's actually what we will measure, the rate of water uptake. Now, we are assuming that the water's being taken up because we're assuming water's being lost by transpiration. But that's not 100% right, because if you think about it, actually the cells in the leaves are photosynthesizing and they need some water for photosynthesis. So we need to bear that in mind. And also some of the water that's taken up by the stem is used for turgor, to make the cells turgid in the stem to give support. But you need to be aware, yes, photosynthesis needs to be remembered, and turgor providing support in the stem. But really, we're assuming the bubble potometer measures the rate of water uptake, which gives us an indication of the rate of photosynthesis. The faster the bubble moves across, the faster water's being lost from the leaves by transpiration. Now, if you were going to use this apparatus, you would record the initial position and the final position, calculate the change in position, and then divide by the number of minutes that you're letting this apparatus run for. Thinking back to the diagram, we can do that as it is, and that's basically in dry conditions, and then we can repeat the experiment by when we tie a plastic bag around it. So if you tie a plastic bag around that and let it sit for a while, the water vapour that is produced by transpiration will be collected in the plastic bag, and that will give you a high humidity. So you just do it low humidity with just ordinary air, and then do it with plastic bag, and that would be conditions that would have high humidity. Now, temperature is a factor. So to look at the effect of temperature, basically you would repeat the experiment, and yep, in a laboratory you can change the temperatures. So you would just repeat the experiment at a range of temperatures. Wind speed. We said you'd expect there to be a faster rate of water uptake if there was more wind. So how are you going to create wind? Well, I mean, how do you dry your hair? You use a hairdryer. We can't use a hairdryer in this case because, okay, you could use it at different settings and get different wind speeds, but you'd also get different temperatures because a hairdryer has a heater element. So you couldn't change temperature and wind speed at the same time. That wouldn't be a fair test. So we can't use a hairdryer, but what we can use is a fan. And fans usually have different settings or different speeds. So that's going to do there. We talked about humidity. And we said actually the more humid the air was, the slower the rate of transpiration would be, and therefore the slower the rate of water uptake. But how are we going to create more humid conditions? Well, if we go back, anytime you do experiments, try and get reliable results. How do you get reliable results? Well, it's all to do with replication. Results are reliable if you were to give the instructions to someone else and they were to carry out the experiment and get similar results to you, then you know that your results are reliable. So replication, and that means repeating the experiment. So if you did it at 10 degrees Celsius and 20 degrees Celsius and 30 and 40 and 50, you would have to do it maybe 3 times or more at each of those temperatures to get reliable results. We said that surface area, well, we didn't actually say, but surface area, if you had a leafy shoot with lots of leaves on it, and compared it to a leafy shoot that looked like that, I'm sure most people would realise this has a greater surface area and this has a smaller surface area. So the one on the left, A, is going to have more stomata. Therefore, you would imagine that it would lose more water vapour by transpiration. So we would say it would have a faster rate of transpiration. How could we set up an experiment to investigate the effect of leaf surface area on transpiration rate? Now, this is what page 16 is asking you. Well, what you would do, happening here, your leafy shoot, okay, and you would put it into the potometer. And there's the bubble. And this is all water in here. Here we go. And we've got a scale over here somewhere. So you set up the apparatus, as it is in the diagram, with a leafy shoot with quite a few leaves. Okay, you would, so that's number 1, set up the apparatus. Number 2, you would record the initial position of the bubble. And then I would leave it for, say, 5 minutes. And then I would record the final position of bubble. Now, you might say after 10 minutes or 20 minutes. That's okay, so long as you decide that it's going to run for a certain period of time. Pick a sensible time. Okay, so we've done it, and in this case we've got 1, 2, 3, 4, 5, 6, we've got 8 leaves. So then what you would do is remove a leaf. And you probably let it sit for a minute or two. You're going to have to reset the apparatus. In other words, you're going to have to reset the bubble to 0, because if we look at the apparatus, the first time we do the experiment, the bubble is going to move in that direction. It's going to move over towards the plant. So say it ends up there, and we record the distance it's moved. In other words, we take the initial position from the final position. If we're going to do the experiment again, our bubble is going to move off the scale, and that's not going to be any good. So what we need to do, in the diagram we looked at first of all, it had a syringe there. So we would use a syringe to return the bubble. If once you press down the syringe, the water's going to go in, it's going to force the bubble back to where it was. Some don't have a syringe, they have a reservoir with a tap on it. So instead of a syringe, that you might see something like that. So in this case, if it's labelled as a reservoir, you would say you would open the tap to allow the water from the reservoir to move the bubble back to 0. And then you let it sit for a few minutes so the plant gets acclimatised, and then you would repeat steps 2 and 3, the initial position of the bubble, leave it for 5 minutes, record the final position, and then you would keep doing that with fewer and fewer and fewer leaves until you've got no leaves left. So that's how you would investigate the effect of leaf surface area on transpiration. Now, one point we need to mention, transpiration rate. So when you see the word rate, another word you probably think of would be speed. So in maths, you learn that speed is distance over or divided by time. So the transpiration rate, it's going to be the distance moved by the bubble divided by the time, which if we did what we wrote in the previous slide, it's going to be 5 minutes. So you divide the distance moved by the bubble by 5. Okay, that's us for now.